Okay, so we're going to go ahead and move in to a keynote presentation for this afternoon, and then you'll get a chance to talk more, I promise. We'll take a little break, and then we're going to have our final session of the day. So it is my pleasure to introduce Dr. Nancy Johnson from it's not, I guess, my alma mater, but where I did my PhD at Northern Arizona University. Nancy is a Regents Professor of Soil Ecology at NAU. She earned a PhD in Ecology and Plant Pathology from the University of Minnesota and a master's degree in Botany from the University of Wisconsin. Johnson and her students study interactions among communities of plants and soil organisms and natural and agricultural ecosystems throughout the world. They have discovered that resource availability influences mycorrhizal symbioses and associated below-ground microbiomes, and they develop empirical tests and theories to better understand these responses. So I think this will follow nicely from discussions earlier today that were a bit more bacterial focused, so here we'll have we'll get the fungi. I've heard you can say fungi or fungi, but you know, tomato, tomato.
There's even fun gals.
Fun gals, exactly. I wasn't going to use that.
I just want to thank Joey and thank Stephanie for the opportunity to come to this symposium and share some of my research. I really am so enthusiastic about getting more involved with arid land agriculture. As you'll see from my talk, I actually have done a lot of work in mesic land agriculture. And so I do have a project now in Maricopa, and so I think going forward I'll be doing a lot more arid land work. I also realized that the title of my talk that I sent to Stephanie was Mycorrhizae in Agriculture, Past, Present, and Future. And somehow that didn't get on my first slide here. But so I want to cover three things. The past comes in with mycorrhizae are incredibly ancient symbioses, and they help plants adapt to their environment. The second point I want to take, it looks like I just turned everything on, oops, is that agriculture inadvertently selects for less mutualistic mycorrhizae. And the last point is I want to discuss the potential for agricultural management to select for increased benefits of mycorrhizae. And the word mycorrhiza literally means fungus root, and it involves a plant that produces sugars and lipids that trades those resources to a fungus in its root, and in return the fungus delivers essential mineral nutrients and better access to water, disease resistance, and resistance to salinity. This is a really nice cartoon, I think, that shows it even more clearly. The plant delivers sugars below ground to its hyphae. The hyphae come in and deliver these resources. And what you see, the orange and yellow lines are thin hyphae on the roots, and it's been estimated that the presence of the mycorrhizal fungi increases the surface area hundreds to thousands of times. Here's some photographs of arbuscular mycorrhizal fungi. We used to call it vesicular arbuscular because they form these vesicles, and the vesicles are kind of like storage units of the fungus. And the arbuscules are little tree-like structures where the fungus exchanges those resources with the plant. And we know that they're very abundant in dryland agriculture. And from a long, deep history perspective, these symbioses predate plant roots. So it's estimated that the earliest land plants arrived about 460 million years ago. They were just kind of like ancestors of algae, and we know that mycorrhizae were around then, and it's pretty clear from fossil evidence. And here's and we also know from the fossil record that the first land plants did not have roots; they just had below-ground stems, and these fungi were in the below-ground stems. It's pretty clear. So again, mycorrhizae predate roots, and these fungi have been around since the very, very beginning. So we could kind of think about it that roots evolved to house some mycorrhizal fungi. Here's just some of that evidence. Here's a photograph of a spore that was fossilized about 460 million years ago. It's dated to the Ordovician, back when there were mosses and liverworts. And here's a spore of, we now call it funneliformis, but we used to call it glomus mossy, and looks pretty similar, doesn't it? Here's a fossil from Scotland, dated at 400 million years ago. Here's a cartoon of what the plants looked like back then. They didn't have roots, they just had these below-ground rhizomes. And looking closer in those rhizomes, you can see these structures, that look like this. And here's a root, a corn root from my lab, very similar structures, hyphae and vesicles. And the arbuscules, there's even fossilized arbuscules on the left. That's a 400 million year old fossil. Here's present day. So again, these things have been around a long past. And it's very likely that arbuscular mycorrhizae have always been present in agroecosystems. So farmers have inadvertently been selecting for mycorrhizae while they were selecting for their crop plants. And I just want to throw in a little personal, my background. I come from a long line of Swedish farmers. This is my great-great-great-grandfather. He left Sweden and settled in Minnesota and became a very successful farmer. So my family still farms today in Minnesota. We don't worry about water. Actually, we have too much of it sometimes. So here's a cartoon showing the mechanism by which arbuscular mycorrhizal fungi help plants overcome resource limitation. You can see this cartoon. On this side, there is no extension of the hyphae out into the soil, just root hairs. And here shows how much hyphae extends into the root, into the fine spots, and really helps get resources. It's quite sizable. All mycorrhizal researchers, we have our favorite big plant, little plant picture. I took this picture in 1983. It's Panicum virgatum, growing in taconite mine tailings. The plant on the left didn't get any mycorrhizal fungal spores. The plant on the right got Glomus tunicatum. I love it. It's just what we always wanted to see. And it's what we often hear about when we talk about mycorrhizae. Everybody loves mycorrhizae, right? They're mutualists, they're good. But the problem is that I did some more work in this same experiment. I did some phosphorus fertilizers. And what I showed was that sometimes the costs of the symbiosis can outweigh the benefits. And the plant on the right is still the mycorrhizal plant. The one on the left is non-mycorrhizal. It happens to be bigger, and I gave it a lot of phosphorus fertilizer. So in very high phosphorus soils, the plants could actually not do as well with mycorrhizae yield-wise. So that's something that I've pretty much been grappling with for the last 40 years. So that brings me to the second point. Agriculture can inadvertently select for less mutualistic mycorrhizae. For my PhD dissertation, I did some studies at 2 agricultural experiment stations, one in YCEDA and the other one in Lamberton. And these were crop rotation studies. And we looked at the communities of arbuscular mycorrhizal fungi in long-term maize and long-term soybean, and how rotation changed those communities. Back then, this was back in the late 80s, we didn't have molecular techniques, so we just floated the spores out in sugar water, and I identified them with a microscope. And we found that there were very different communities in corn and in soybean. And so essentially, in the corn, there were a handful of Glomus species that were always more abundant. And similarly, another group in soybeans that was always more abundant. And so I took my data and looked at the relationship between the populations of these proliferators and the performance of the crops in those same plots. And I was shocked and dismayed to discover that the groups of fungi that seemed to proliferate in corn were strongly negatively correlated with the performance of the corn. It had lower yields and less tissue, phosphorus, copper, and zinc. And those are all nutrients that we know that the mycorrhizae takes up. It was symmetrical. Soybean was the same problem. Come on. I don't know why. Can you make it advance to the next slide for me? Thank you. There. I can't imagine you are. Anyway, same thing. The taxa that increased in the soybeans were highly negatively correlated with the performance of the soybeans. So that's not at all what we would expect if this is a wonderful mutualist in all places at all times. So here's where the 500-pound gorilla comes in. This is my favorite gorilla. His name is Richard. He lives in the Prague Zoo. And he's just become part of my life. If you're interested in learning more about Richard the gorilla, just do a Google search. He just turned 30, and they have some lovely videos about him. But at any rate, here we have agricultural management can inadvertently select for mycorrhizae that reduce crop yield. So that became clear during my dissertation work, and so we published this in Agronomy Journal in 1992. And as you can imagine, it wasn't a big hit because it's not what people want to hear. At any rate, more recently, there was a paper published in New Phytologist by Megan Ryan in Australia and Jim Graham from Florida. And they summarized the last 20 years of evidence to suggest that sometimes agriculture can create less beneficial mycorrhizae. And their title, Little Evidence That Farmers Should Consider Abundance or Diversity of Arbuscular Mycorrhizal Fungi when Managing Crops. So as you can imagine, this caused quite a stir among mycorrhizal researchers. And then it was followed by some letters and forum suggesting why farmers should manage arbuscular mycorrhizal symbioses. And these are my friends too. All these folks are friends of mine. I agree with both papers. If we're looking for yield and we're working in high input agriculture where there's plenty of fertilizer, don't manage for mycorrhizae. You don't need to. But if we're looking for management of soil properties, soil aggregate stability, resistance to diseases, resistance to salinity, then do. And so that's, I think, where we're at. We just have to be smart about it. I had a wonderful opportunity to work with some researchers in China who have done a long-term experiment with tillage and fertilizer, 14 years of either no-till or conservation, or no-till or conventional tillage with a mold-bore plow, and either no fertilizer, nitrogen only, phosphorus only, manure, both nitrogen, phosphorus, mineral, or all three. And this was done for 14 years before the 3 years of data collection. So it's really a nice, very robust, replicated experimental design. And it was really informative. It pretty much backed up both the two papers that I just described to you. We have a problem in mycorrhizal research because it's like Joni Mitchell, you don't know what you've got till it's gone, right? So there's no way to know what mycorrhizae are doing in the field because they're always there. So to figure out what they're doing, we actually have to take some soil from the field, bring it into a greenhouse, remove the mycorrhizae, and then either put it back or don't put it back. And so that's what this slide shows. These data show, this was winter wheat. This is the mycorrhizal growth response. So we grow a plant with or without its living soil inoculum. And if the biomass ratio is above this dotted line, that means that it was a mutualism. It was beneficial. If it's below this line, it caused a growth depression. So the green symbols are no-till, pink is conventional tillage, and you can see that only the unfertilized control treatment actually had a mutualistic relationship. All of the conventional tillage treatments were below the line, and if there was only nitrogen, no phosphorus, it was neutral. So we can conclude then that tillage and fertilizer creates AM symbioses that reduce the winter wheat biomass in the greenhouse test. But we could also go out now with molecular tools. We don't have to look at spore communities anymore, thank goodness. And with the molecular tools, my colleagues were able to identify over 77 different virtual taxa of AM fungi. And what you can see is that conventional tillage significantly reduced the diversity of arbuscular mycorrhizal fungi, pretty much across the board. And here's another illustration of the changes. The list here, these are virtual taxa with their special numbers, but the next, the abbreviations have to do with the genera, Chloridioglomys and Funneliformis. They both increased in the response to tillage, and Rhizophagus and Dominica genera decreased. And so clearly, farmers are inadvertently selecting for different communities of fungi. And how do those different communities perform in the crop? So again, we have, I've just been so interested in the discussions today about the importance of holistic perspectives and using more holistic analyses. And one of the ways that we do that is with structural equation models. It's just a way of taking a lot of factors and linking them with causal relationships with how we hypothesize that things link together. And so these are a couple structural equation models that we did and fitted with our data to look to see what factors best explain this variable. It's called soil multifunctionality index. And the multifunctionality index is a kind of a holistic index of lots of different variables related to enzymes and things that have to do with nutrient cycling. So this is what people want to have happen in sustainable agriculture. This is a really important index. And what you can see is the blue is actually a negative relationship, think cold. So tillage reduces the soil multifunctionality index quite a lot, but it increases, as I mentioned earlier, this Chloridioglomys, and fertilization also increased Chloridioglomys. If the lines are dotted, it's not significant. But these two fungi that decreased in the tilled plots, you can see that Rhizophagus actually is significantly positively correlated with this variable that it indicates it's more sustainable. So it's complicated. And at dinner last night, Joey mentioned to me that his advisor told him, never say it's complicated. The goal of scientists are to make things less complicated. But it really is complicated because mycorrhizae in production agriculture, unless we really want to cut back on fertilizer and have cut back on fertilizer, it's probably not going to increase our yield. The way to increase yield is probably with fertilizer. But if we want to use some of these other services that you can get from mycorrhizae, then we really want to focus on understanding the mycorrhizae. And so this is the last part of what I want to cover today, and that's talking about the future, thinking about the potential for agricultural management to select for increased benefits of mycorrhizae. And in case you don't recognize that, that's a colored image of our hero, Charles Darwin. So again, I mentioned that arbuscular mycorrhizae have been shown in field trials to be used as biocontrols against fusarium wilt. And there's quite a lot of publications showing that. Also, they have been shown to increase the tolerance of crops to salt. And so these are the kinds of things that we should be focused on. So how do we start to develop methods to select for beneficial traits in mycorrhizae. And I've done a lot of work, although agriculture is half of my focus. I actually have studied mycorrhizae in natural ecosystems all over the world. And one of the questions that I'm really interested in is what does it take in these local areas for the mycorrhizae and the local plants to become locally adapted. And we see evidence of local adaptation in many different experiments. And it just essentially means that organisms do best in their home environment. And it's not just that they do it best in their home climate and their home soil, but they also do best when they have their local communities of microorganisms, mycorrhizal fungi and bacteria. And I'll just share with you one of our experiments that show this. The terminology we use is sympatric if the organisms are coming from the same place. And so sympatric organisms are adapted to each other. And one of our graduate students a couple years ago published this paper where he took Bouteloua gracilis, that's blue grama. Those of you that are from northern Arizona know blue grama. It's a wonderful native grass. And he collected seeds and soils from an elevation gradient where it created, it wasn't a very far distance, but because of the elevation, we had wet and dry populations. So he got two seed sources, one from wet, one from dry, and he had their soil communities, and he did what we call a reciprocal inoculation, where he grew the blue grama with their home and what we call away combinations. And then he slowly let the plants die of drought. And he watched the death process. And it was quite clear that the plants that were grown with their home mycorrhizae and other microorganisms, there were bacteria in there too, but we just didn't measure those. They were clearly much better able to deal with drought. They lived longer, they grew bigger. Everything was clear that they had an adaptation with their local microbes. And we've done some climate change work where the work was actually done in the field, and it clearly shows that locally adapted microorganisms make their host plants more resilient to climate change. We have just got a paper in climate change biology that shows that very clearly. So yeah, we've shown it in the greenhouse, we've shown it in the field, that if plant genotypes have their home microbiomes, they can withstand a warmer and drier environment. If they don't have their locally adapted communities, they don't do as well. So we've been thinking a lot about the process. And I know when I first started talking with Joey about collaborating on some research, of course we would think about, well, let's develop, some lines that we could, use for inoculum. And I've come to the conclusion that using mycorrhizal fungal inoculum, especially in production agriculture, is like sprinkling salt in the ocean. It's just not the way to go about things. But what we can do is we can try to mimic natural selection through our artificial selection and select for the most beneficial symbioses in situ. And that is cost effective. But we just have to know what we're doing to do it. And so plants and mycorrhizae can be thought of as complex adaptive systems. And in natural systems, there's a reciprocal selection, feedback. The plants do better when they have a microbiome, not just mycorrhizal fungi and bacteria, but also little animals, nematodes and mites. The whole thing can be reciprocally selected. So yes, there's going to be pathogens in there, but then there's the enemies of the pathogens, and you have a whole system that's evolving together. So with this reciprocal selection, you get these feedbacks that can be stabilizing over time. So evolutionary feedbacks then occurs when natural ecosystems and the whole plant can drive the selection. And inadvertently, when us farmers go in and put in our crops, we don't usually choose the varieties of our crops to match the microbes in the soil. We don't really know what to do, and so that's something that we might want to start paying attention to. Also, we add plenty of fertilizers and essentially make the mycorrhizal symbioses redundant, or even increase the costs relative to the benefits. So future research then is important so that we can develop agricultural management that will encourage the more beneficial mycorrhizal symbiosis and discourage the costly ones. And so to manage for increased mycorrhizal benefits, we should select for mycorrhizae that increase the resistance to disease, drought, and salt. We should select for mycorrhizae that can improve soil structure and nutrient retention. And finally, we should select for mycorrhizae that can maintain yield under reduced fertilizer and tillage. And that's something that we really want to pay attention to, because as phosphorus fertilizer gets more and more expensive, we might have to cut back on phosphorus fertilizers. And if we could understand the mycorrhizal system well enough, then we could select for the most beneficial symbioses. So I'll end here. This is a photograph of a Maricopa. That's a Maricopa mycorrhiza right there. There's a lot of them there, and I'll end here for any questions there might be.
So thank you for the presentation. It's a great presentation. I truly enjoyed your presentation. My question is that we found some fungi as well for the Fusarium heptili in cereal crops and in drought resistance as well. So what is your recommendation for treating the fungi for big seed? Is it seed treatment is okay, or just to use the spray?
You mean for adding the mycorrhizal?
Yeah.
I don't think, at this point, I don't think we know. I know that there are inoculum products out there. I would go back to the panel this morning and do a large-scale field trial in your system and find out whether or not they work. In my own experience, we haven't yet found industrial-sized mycorrhizae that actually I would recommend putting in the field.
Great. Then just last question, sorry.
I wish I could be more positive.
FDA is not approving that instead of using sprayer, that you use just seed treatment, and then how easy to support production and everything that you need to list. So that's why I was asking your recommendation.
Yeah.
So let's say, assuming that we found one fungi that is like treating two traits, for instance, both Fusarium and as well as the drought.
Right.
Do you think that fungi is trustable, or like over the time it's going to be just mutated, or anything happens? What is your recommendation?
I am so glad that you asked that question. Over time, will it change? And I bet it will. I think that's the problem, that right now it's such a complex system. There's so many players that one year it might work very well, and another year it might not. And so these products that, you know, they need to be tested in situ, and then go big and keep watching. The problem is a lot of those mycorrhizal fungal products aren't really what they say they are. And they also need to be carefully screened for the presence of pathogens. And the third thing is sometimes they are not anything at all alive, it's just fertilizer. I know. But so we have a long way to go. But I think there is great potential, but I don't have answers, because I think we still need to do a lot more research. And this is where I think we could get more and more people involved in partnerships with growers as these technologies with the molecular genetics get more and more developed. We could probably, with some of the talks we heard this morning, we could probably match up, you know, some of the genetic analyses with the genomic analysis and figure out who is where when, use some of these more holistic ecological models like structural equation models, and then decide, okay, it looks like it's an El Nino year. I think, at least where I live, that means a lot of snow. I don't know if it's raining down here, but we think it's going to be cold and rainy or whatever, and I think we're going to have more fusarium or whatever. So there's a lot that needs to be done, but I think we're finally seeing the tools are coming together.
Well, yes, just your opinion, do you think like there's like a better strategy to favor the symbiosis is like improving the soil conditions to make the mycorrhizae to prosper, or do you think there's also a lot of room to work on the plant side? So I'm coming from a lab, you know, my PhD, we were studying this a lot about like strigolactans and the emission of them in the root exudates. So can be a strategy also like to enhance the plant to release this factor, like this signal for the mycorrhizae to colonize the roots. And yeah, in that case, it's like a little bit like more biotech, but or metabolic engineering, if you want to call it like that, but that's also a strategy, I think.
I couldn't agree more. If you didn't hear his question, he asked, we can work on the fungal side, but also why not breed for crops that are better hosts and actually benefit more from the mycorrhizae. And I couldn't agree more. And this has actually been, I think, part of the problem of mycorrhizal yield effects is that where we have our seeds and our development in these big farms that are developing varieties, they are fertilized completely. There's no water limitation, no nutrient limitation. So most of our crop hybrids and that are in production have been selected to be non-mycorrhizal, even though they come from the C4 grasses typically are highly dependent on mycorrhizae in the wild. So now that we know this, I think we can pay more attention to the genetics of the symbiosis and try to select for crops that can actually harness biological soil fertility in the arbuscular mycorrhizae, in the nitrogen fixers that might live with the arbuscular mycorrhizal fungi. We can actively select for crop cultivars that are more likely to use biological fertility instead of chemical soil fertility.